The dream that led to tissue engineering was the idea that we could, instead of making this, we could make living tissues to go in to your body. And the first paradigm, the first idea towards this was to take cells from people and put them into 3D polymeric artificial scaffoldings that look roughly like the tissue shape you wanted, and hope and work and engineer at growing that up over a long period of time into the tissue you wanted. This has turned out to have quite a range of limitations, because effectively you're indirectly engineering. You're hoping and working on the cells to do things for you, and the controls aren't very good for that reason, and the speed, of course, is very, very slow. It's a cultivation process. So we found that we could stumble on a means to do what is something of a dream, really, to make a vision of making tissues like you make mobile phones, engineering them directly. And this comes from a material which is this one here, which is collagen. It's a protein that makes up the majority of your tissues, and your cells are embedded into it. It's a strong protein, makes about a quarter of your ligament and tendon and so forth. And this is what it looks like when it's been made into a gel in the laboratory with cells embedded in it. As you can see, it's pink because of the culture material, but it's extremely weak. In fact, it's 99% water. You can see the water's oozing out of it already. But if I place it on here, and this was an accident we stumbled on in the lab some years ago, doing something a really strange thing in effect is to compress this. So if we put the blotting paper on the bottom and a weight on the top, you end up with a compression system where the fluid is forced very rapidly out of that collagen material from around the cells. Surprisingly, the cells aren't damaged by this. And what you end up with is something very dense, controllably dense, like these here, sheets of, in fact, living tissue. This is natural protein, which is what you're made of, and your cells, if that's what is placed in. But in this particular case, the process is pretty well completely controllable, because we control how much fluid goes out there for how much cell density, how much protein density, and because of the direction, you get very nice biomimetic tissue-like stacking of layers as well. They are thin, though, you can see here. And if you're making a complex tissue like skin, this actually turns out to be an advantage, because it's different further down and changes all the way up. So we have now this concept of building up tissues by layer engineering, by stacking them. So instead of spiraling these together, which is what we've done with these two pieces here, we can stack them on top of one another, and make tissues which are different as you go up. Each of these, of course, would be made intentionally, rather like a computer-generated material, so it could be made very simply differently. The first clinical application of this, because it's thin and transparent, is likely to be with the Institute of Ophthalmology, with work on the corneal transplantation, where stem cells are placed over a burned cornea, and they'll be carried on this collagen, which is made in front of the patient in the operating theatre. And here we get the clue to why this is such an exciting paradigm shift, we think, in our eyes. This is an automatable process, and we've been working for some while now with a company called TAP, and they've made this machine which effectively compresses many, in this case 24 of these pieces of tissue all in one go within 45 minutes. So instead of this happening in days or even weeks, with lots of culture and not much control, it now happens with very high control, but in a very short period of time. Potentially, of course, at the bedside. So now, instead of having a long process outside the theatre, this could all happen next to the patient. What's more, and this is what this machine is designed for, these are now ideal for testing materials, for testing, replacing animals, for testing pharmaceuticals, for screening chemical toxic agents. And this could effectively save thousands and thousands of animal experiments in the early screening times, simply by making hundreds of these tissues and screening your agents on them. So this, incidentally, is what a series of those looks like when they come out. So many, many identical tissues. And this is the point. They're all much better than animal tissues in that way. They're absolutely identical. And they're made in this manner here. This is the primeval way we did it originally. And you can see now the tissue is left compressed onto there, very, very thin, but dense at the level that we calculate it to be. And you just either spiral it like this, or in fact layer on, stack on stack on stack.